Hello, everybody. Pleasure to be here, and thank you for the invitation. So I will take you through proteomics as we use it in obesity for molecular phenotyping. And I'd like to start out with an outline of my talk. I will first introduce a concept that you're very well familiar with, that basically the phenotype is a result of the interaction of the genotype with the environment. And then I will introduce what we understand under the term molecular phenotyping. I will talk about clinical proteomics, our platforms that we have developed. Also show you how you can basically execute proteomics inspired by metabolomics platforms that we are also using. I will briefly talk about alternatives to mass spectrometry, which is our main workhorse, and that's to say introduce aptamers. And then I dive into the main subject of this talk, which is using systems biology approaches in metabolic health and presenting you a large clinical study in obesity. So when the genes meet the environment, the genotype expresses a molecular phenotype. And you can see here on the left-hand side, our epigenotype and genotype. On the right-hand side, diet and lifestyle factors. And in the middle, you have the evolving molecular phenotype at proteomic, metabolomic, and also at nutrient level. So we have on top of the diagram here what results as physiological or pathological processes that are derived from environmental cues and from your heritage and your development. And if we all were computer scientists, we could say we have on the left-hand side the hardware and the software, on the right-hand side the user input, and then we have a graphical user interface, so to say, to interrogate what's going on at molecular level. So we are using different platforms, omics platforms here, and my focus will be on proteomics. This is just to say that these omics change across the organization of an organism. So you can look at organelle, cell, organ, or whole organism level. Of course, they change over time as we age and as the environment changes. And these analysis levels are very challenging to be integrated. Now, let me just spend a few minutes on clinical proteomics. What does it mean in practice in terms of proteome coverage and analysis time? So the ideal sample you would take is minimally invasively accessible. So we talk about blood essentially, or urine also. To some extent, you can also look at cerebrospinal fluid, CSF. And the ideal sample would give you many thousand proteins in a very short time. However, the reality is different, and I've cited here a few publications from the last years where thousands of proteins have been analyzed, but it took quite some time to acquire all these data. And if you look at this diagram here about properties of these workflows, you can see here that traditionally these proteomic workflows are very good for proteome coverage and quantitative performance, but less optimized for low cost, low sample consumption and throughput. So we have developed several workflows and applied them to several studies, as I'm displaying it here, where we still see a very high amount of proteins, but we can do this in a much shorter time. And we also look at other optimization parameters. So we have developed these blue workflows here, where we have, as we think, a good compromise between still a good proteome coverage and very accurate quantification, but we enhance the throughput, reduce the cost, and reduce the sample consumption. Now, this is the practical platform and workflow we have applied. So you start with the samples, plasma samples. You filtrate them, you do multiplexed immunoaffinity depletion to get rid of the most abundant blood proteins. You do some buffer exchange, you apply tryptic digestion, and then you do what we call isobaric labeling. So you use chemical reagents that put tags onto the peptides, and then you separate this by chromatography and directly infuse it into mass spectrometry. And to assess the reproducibility and robustness of our workflow, we have prepared here 96 individual samples. They were all identical and they were all labeled individually. And what you would expect is a theoretical log 2 of 1 which means all proteins should be there in the same quantity because it's all identical. And this shows you a very narrow distribution around this log2 of 1. And this tells you that every signal, every fold change below 0.77 and above 1.3 can be regarded as a true biological signal, which is very good. And then in terms of linearity, we spiked proteomes, known proteomes or combinations of proteins into real blood plasma, and you would expect a linear correlation. And this is exactly what we see in practice. Now, when you do classical proteomics, you do basically first a protein identification based on peptide sets, and then you quantify these peptides. And we tested the performance of the classical proteomics workflow on very large data sets, and you will come across the term Diogenes. This is a large European study to which I will refer in the biological part of the talk. And we also looked into cerebrospinal fluid data sets. And here are just a few numbers that should tell you that these amounts of data and spectra is very, very big. So if this is the total of all spectra you would acquire, that doesn't mean that the spectra all translate in protein identifications. So when you do first identification and then you quantify the identified peptides, you typically see this, that about 1/5 of the spectra yields protein identification, and then about 17% of these spectra actually yield not only identification but also quantification. However, when you change the mindset and be inspired by metabolomics, which is largely based on NMR spectroscopy and more recently also on mass spectrometry, we tried actually to do it the other way around. So first we quantified all the peptides regardless of what they are, and we did a differential display as you would do in metabolomics, and then we identified these peptides with post-translational modifications, and you can clearly see here a graphical illustration that you gain a lot of information because more spectra yield information. And this has been quantified in these large data sets I've been alluding to before. So on the top of this table, you have CSF data sets, sample sets, and on the bottom, you have the plasma ones. And we compared the classical to the QTI workflow, which means quantification and then identification. And you can clearly see an enormous increase in quantified peptides and still a very significant increase also in quantified proteins. So this encouraged us. However, mass spectrometry is not the only workhorse you can use in plasma proteomics. There are also affinity reagents, and I introduce here the SomaLogic technology, which is based on DNA aptamers. These are basically small pieces of DNA that work like affibodies, like small antibodies. So the proteins bind to these aptamers, and then you have the advantage that you can amplify the signal because you have DNA bound to the protein, and then you can do identification and quantification. So this gives you kind of a PCR for proteomics that you usually only know in genomics. However, it's an indirect measurement of proteins. You can get thousands of plasma proteins at a very high throughput, and it's very sensitive. However, we found it to be less precise and less accurate than mass spectrometry, which in our view is very complementary. You go from proteins to peptides, as you have seen, you separate them, you sequence, identify, and quantify, and that represents a direct physical measurements of the peptides. However, you typically in plasma, because it's so complex, you end up with hundreds rather than thousands of proteins. It's lower in throughput, it's less sensitive, but it's more precise and more accurate. So what we have done in our laboratory, we combined the best of both worlds, the MS specificity, precision, and accuracy with SomaLogic sensitivity and throughput, and that's actually our recommendation. Now, a few years ago, we published a perspective on a systems approach to diabetes research, and this, of course, extends to the whole metabolic health area. And we reviewed human type 2 diabetes studies from a genetic, epidemiological, and clinical perspective. And we know that all complex diseases are multifactorial and arise from these gene-environment interactions I have introduced. However, type 2 diabetes is also such a complex disease, as we know, and it's a collection of subphenotypes, as previous speakers already mentioned. So this is why we propose and applied a systems biology approach with new clinical study designs. These study designs are longitudinal, for example, where each subject can serve as its own case at control. And these subjects are not only studied at homeostasis, but also before and after safe challenges, for example, metabolic challenges. And then everything is based on omics integration and deep phenotyping. So this is how such an integration map can look like. This shows you such a graphical display of a systems approach to analyzing complex traits. So we focus here on data from accessible tissues that can be adipose or muscle tissue or other biopsies, and body fluids like plasma and urine. And since the measured molecules, proteins and RNAs and metabolites, are all influenced by genomics, DNA must also be characterized, of course. And then we have epigenetic events like DNA methylation, microRNAs, and histones, and they also change genomics and can be measured in some tissues. And these regulators are in turn regulated by these environmental signals of nutrients and bioactives in food, for example, by physiological factors and personal and public environmental conditions. So these metadata must then be captured in next experiments to understand the context in which they are captured. The challenge is, of course, integrating these multi-omics data sets. So this is another busy slide, but don't worry, I take you through this and explain it to you. So this is a network of biological molecules causally linked to type 2 diabetes, to the type 2 diabetes molecular phenotype. We call this a biological expression language framework. So you can see here node colors, and that tell you, these colors tell you the molecule type and molecular processes. The red links are molecular interactions, and the gray links are process molecule causal relationships. So this network is constructed by extracting all nodes in the BEL knowledge base, and we focused on terms insulin secretion, insulin receptor signaling, glucose transport, glucose import, and glucose homeostasis. And this network, as you can see, contains molecular nodes, biological process nodes, as well as causal relationships. So this basically gives you a map of which factors are influencing type 2 diabetes in which way and at which level. So now I come to the core to the talk, and I would like to introduce you to the Diogenes project. Diogenes stands for Diet, Obesity, and Genes, and has been a large multi-center, multidisciplinary European research project to advance the understanding of how obesity can be prevented and treated from a dietary perspective. The design of Diogenes was first a caloric restriction introduction phase, as you can see here, between CID1 and CID2. So obese subjects were put on an 800 kilocalorie per day diet. Actually, I was happy to be not involved in that study, so that's very little per day. And we all know that when people adhere to these 800 kilocalories, they will lose weight no matter what. And then the decisive point here is that we have a weight maintenance phase here between CID1, which stands for clinical intervention day 1 and 3. And we focused here on 500 of these initially recruited more than 1,000 subjects. And these 500 were chosen because they could at least lose 8% of the body weight during caloric restriction. And then we looked at them also post-weight maintenance. And we measured blood plasma samples from those 500 subjects at 2 time points. And we applied the proteomic workflow that you have seen before. So we have about 1,000 subjects in total, 4,000 samples, about 7,000 variables. And then the weight maintenance phase was considered 5 different diets, and they were differing in glycemic index, in protein content, and we had a control diet. Now, the first question was, And the key question was, can we predict success in weight loss and maintenance at baseline? So when you recruit subjects, you do a proteomic analysis, a fingerprint, so to say, can you predict whether they lose weight or not on which diet? And the answer is actually yes, we can. As you can see here on the ROC curves, where different percentiles are displayed, and the diagonal line between sensitivity and specificity would be just flipping a coin. So that's just random. And you can clearly see here the area under the curve is very nice, and that tells you that our proteome profile that we captured at baseline can be actually used as predictors for successful weight management. Now let's look into the proteomic study in more detail. We published this, and you can see here as a first insight a volcano plot where quantitative plasma protein ratios between these CRD1 and CRD3, that means baseline and post-weight maintenance, are displayed. These data concern all centers involved, all different clinical centers, and across both genders. And interestingly, most proteins that we found regulated in our nutritional intervention were actually reported to be associated with weight loss in obese individuals. So not only are they predictive, they're also functionally meaningful. And the red proteins are up regulated, the blue proteins are down, and those proteins in bold have post-mass spectrometry confirmed by SomaLogic. Here you see a heat map of plasma proteins differentially expressed between these clinical intervention days. And those proteins significantly correlated with the increment of selected clinical variables. As you can see here, BMI, the HOMA index, the Matsuda index, and the fat mass. And again, this is between CRD1 and 3. And this is an example of single centers. The left colored heat map shows you the estimate of the coefficients, and the right grayscale heat map shows you the p-values. Now, we went into something that was at the time pretty much unheard of. We went into clinical replication of these large-scale proteomic results. So how could we do this? In the bottom line, you see here again what we now call the C2A and the C2B data sets. So these are the Diogenes samples and subjects and data at baseline and after weight loss maintenance. And then we had access to a program in obese and overweight patients, a clinical weight management program at the Ottawa Hospital in Canada. So that was our second cohort, and as you can see here, the numbers are quite comparable. And then we did, again, differential proteomic display of plasma proteins in all of these subjects. Now, this is the outcome here. Here you see scatter plots of individual protein standard deviations to give you an idea which proteins naturally fluctuate and which are more stable. And you can see here a comparison between the C1 cohort versus the C2 and C2A and C2B between the two collection days. So the least variable proteins are in gray or a fading color. And on average between the measurements, these are the ones, also the green ones, these are the proteins involved in decoagulation and complement cascades, whereas the most variable from red to orange were commonly quantified between the cohorts. And for example, CRP is among those, which is a standard clinical marker for inflammation. In general, we found less variability in C1 than in C2. This means that the Ottawa population was probably more uniform, which makes sense because it was a regional hospital there, whereas the European study went across different nationalities. And now I show you some more volcano plots about clinical variable protein correlations for C1 and C2A. So again, Europe and Canada. The colored dots the dots are colored if they are significant for one cohort, and the colored names tell you if these proteins were significantly found differentially expressed for both cohorts. And here you see two volcano plots for BMI protein correlations. And here you see two volcano plots for insulin protein correlations. And you can see here on the bottom the abbreviations of the proteins that have been found regulated. Again, the red ones are up, and the blue ones, we didn't find so many, were down-regulated. Same principle here. Here you look at glucose protein correlations. So correlations between glucose levels and these particular proteins. And here you can see correlations between plasma proteins and cholesterol levels of the clinically enrolled subjects. Now, this all now led us to a construction with the means I've been talking about before, to a so-called 3 nearest neighbor network. So these are common plasma protein neighbors in both C1 and C2A, so Europe and Canada cohorts, based on our found proteomic relative quantification. So these are protein networks that are known to be functional, and several of these associations have been previously reported as predicted functional associations. And these are the clusters here, A, B, D, E, G, and H. And interestingly, the light blue cluster H comprises inflammatory proteins. So I come to the wrap-up of my talk. We had here 2 populations under investigation on 2 different continents, with about 500 participants in each cohort. We replicated the findings of the European study in a randomized control trial, and that was at the time absolutely new. We found several concordant protein measurements in terms of variability, gender specificity, and age relationship. We replicated known and also found new associations between proteins and clinical and molecular variables, such as body mass index, insulin and glucose, and cholesterol. And we identified protein markers of obesity severity that can be now tested in clinical practice. And we established this association map of plasma proteins of obesity and overweight for functional information. So we believe that these proteomic findings were only possible with this large sample size because of the usual statistical constraints and biological variability. Also remember that we had an enormous number of variables being tested, so you need a large sample size to correct for multiple testing. The practical feasibility and efficiency of this discovery replication with clinical mass spec-based proteomics was delivered, and it shows that these high-tech platforms, once they are automated and very robust, can be used actually for significant and tangible and translatable clinical findings. And in generally, what we have seen, that the high protein and the low GI diets perform better for weight management, weight maintenance. So now you will say, well, we don't need proteomics to find out about that, but that's true. But however, never forget that the inter-individual variability is extremely high, and we found the same thing here. And this brings me back to the introduction of my talk, that it is important to not only look at group averages, but also follow individuals over time, because none of us is an average or a mean. We are all individually very, very different and would follow different metabolic trajectories. With this, I would like to close, and I thank you for your attention, and I'm happy to receive questions.